So today is my greatest pleasure to introduce Professor Jason Yeatman. I have known him before he had even started graduate school, as I was just saying. I think it was 2007 at Stanford University. He was already a really high power rising star as a research assistant at Stanford, and an exceptionally creative and thoughtful thinker back then, and of course now as well. But for students, I think some students, a lot of people, when they think about these junior superstars, and I want to confirm if this is true or not. So I was curious, and I was looking at studying his resume. I love studying superstars' resumes, so I was studying it. And I think when you were applying for graduate school at Stanford University, I don't think you had a peer-reviewed, at least empirical paper published when you were applying. Is that correct?
That's true. That's true. And also, I think it's always good for, as we go at different stages of our career, to admit our shortcomings to it. I think I got rejected from 80% of the grad programs I applied to too. So,
oh, wow, lost for them.
Yeah.
That is an interesting story. So since it's the application and interview season for students, I'm sure, I thought you might be curious that you must have to be a superstar or get into a great program, that you have to have a lot of publications as an undergrad, but that's not always the case. And here Jason is. But now I think I was also counting or looking at your resume and looking at how many you had when you graduated from graduate school. And I think that year you had like over 20 publications with 9 first-time authors. So I think he's an, so that was kind of, and of course, it's not just the number of the publications, but the depth and breadth of publications, I think, and high impact to researchers in our field. And that has been quite remarkable. So he's published widely in a number of different Nature series journals, as well as in graduate school. He already had 2 PNAS articles, among many others since then. And so we look forward to his talk. But just a little bit more about him. He received his PhD in 2014 from Stanford. He worked with Brian Wandel in vision sciences and neuroimaging, and then he was an assistant professor at University of Washington until fairly recently, when he came back to Stanford in 2019 to work as an assistant professor in the School of Education, Pediatrics, and Psychology. As I said, I've known him since we were both at Stanford, but also we have continued to work together in different ways, including working on the scientific advisory board of the International Dyslexia Association as co-chairs. He's also received numerous prestigious awards, including the Early Career Award from the Society for the Neurobiology of Language, and the Science of Learning Award from Flux, which is a developmental cognitive neuroscience society, in 2017, and then Jacobs Foundation Early Career Research Fellowship in 2019. All very prestigious awards. And I'm always so impressed by his scientific curiosity, incisive approaches to his scientific research, and then particularly his breadth, going from, and many of you know his work in diffusion imaging and rigorous methodological research, but he also does a lot of translational work. And he was just telling me that he was in the schools all day in the classroom. And so it's really a tremendous pleasure to have you, and I look forward to your talk.
Well, thank you so much, Miko, and it's really a pleasure to be here. I've been scrolling through the different faces and names on here, and nice to recognize many folks. And thanks for the wonderful introduction and bringing me back to my first days here at Stanford. I guess I will, before I talk, Fumiko mentioned something about, younger folks, research assistants, or people just starting grad school, and, how we confront rejection. And, one of the wonderful things about this career is getting to pursue the things you're passionate in, things that excite you, things that keep you interested. That's what I've loved about a career in academia. And, along this path, there are so many moments of rejection. So for those of you applying to grad school, it is true. I got rejected from, I think, 80% of the grad schools I applied to, and then, got in. And I think at each stage we confront these rejections, and if you love the career, just keep going. I'll also invite you all to interrupt as I'm going. Feel free to throw a question in the chat, and maybe Fumiko, I'll ask you to moderate if there's something that's interesting that comes up. I'm happy to stop as I go. Though if it becomes too much, I'll take control again and come back in. So I'll invite you all, unmute yourself if you want to ask it, or type it into the chat. I'm happy to have this be a conversation. We can all pretend we're sitting in a seminar room together, rather than our bedrooms, offices, and wherever else everyone is. Okay, so let me dive into this. So a lot of people here study literacy, but for those that don't spend their days thinking about reading, I want you all to consider literacy in the context of child development. So I want you to consider the two people in this picture and realize that the two people see fundamentally different things and will continue to for years. As I look at the page of this book, I can't help but hear the rich sounds associated with the printed letters, the language, the story that's told by the author. Well, as my first daughter here, this is actually a couple years old, this photo now, just sees a bunch of pretty pictures. And the interesting thing about literacy is that continues for years. So even a couple years later or a year later, as she could speak, she could demand things of me, like tell me that I need to read her another book. She could communicate and understand spoken language. Yet text was still just meaningless squiggles on a page of pictures. So an important point to remember throughout this talk is that literacy was invented by modern societies. Our brains are not pre-wired to read, meaning that the neural circuitry of literacy is constructed in response to education. That means for those of you that are teachers or were teachers, the work that you do in a classroom, I was in a classroom all day yesterday, is literally constructing the neural circuits that are able to achieve this incredible function. Now, from a methodological standpoint, I think this also means that literacy is the perfect model system for studying plasticity in the human brain. We can do intervention studies, we can do randomized controlled trials, we can manipulate environmental factors in a very controlled way to understand how different aspects of a child's environment sculpt the development of their brain. So let me exemplify this point by comparing what's going on in my brain versus my young daughter's brain as we're looking at this book together. Now, as we open the first page and look at this picture of the bunny rabbit, things start off in a relatively similar way in our two brains. The retina encodes the image here and sends this information back to primary visual cortex, back here at the back of the brain, which starts extracting the most elementary properties of the image that's in front of our eyes. A sequence of regions located primarily along the ventral surface perform a series of operations that are involved in visual recognition. And the first approximation, I think it's fair to say this looks relatively similar in my brain and my daughter's brain as we stare at this picture of a rabbit. However, things are fundamentally different as soon as my eyes saccade to the text here. Things start off the same way, but within this circuit that's involved in visual recognition, a specific region starts computing properties of the text, a specific region that exists in my brain as a literate adult, and is located here on the ventral surface, and is involved in transforming this visual information into a speech and language code. So basically plugging our visual system into our language system to be able to take this visual image and treat it as language. Now, of course, I'm reading out loud. I'm not just leafing through the pages with my daughter. Now, as soon as I start reading the book out loud, we would see activation in these same speech perception and speech production regions, my daughter, to the auditory language. Now, one important point about literacy too is we look at this distributed circuit that requires information flowing from back to front across much of the cortex. The white matter pathways, or the bundles of axons that carry information between these regions, are critical for the development of reading skills. So throughout this talk, I'm going to be focusing a lot on two specific pathways. One is the arcuate fasciculus that's been known to be involved in language and particularly phonological processing. It connects, amongst other regions, Brochet's area and Wernicke's area. It also has projections down to the visual word form area down on the ventral surface. The other pathway I'll be discussing is this one shown in orange, the inferior longitudinal fasciculus. It's a major information highway going from the occipital lobe up to many other regions of the brain. Okay, so 4 points to remember throughout this talk. First is that literacy prompts the development of specialized circuits in visual cortex for rapid and automatic word recognition. Second, in the literate brain, written words are processed like spoken words. You can almost think of becoming literate as plugging the visual system into your brain's speech and language circuitry in a new way. Third, the structural connections between visual and language circuits are essential. And finally, the development of the circuit goes awry in some children, and this is what we refer to as developmental dyslexia. So our approach to studying the development of reading and the neurobiology of the brain is to think about inspiring a virtuous cycle between education and neuroscience. Most of the studies in my lab start off looking like this: intervention or education programs, usually in small groups of young children. And we use these as a tool to understand how a child's experience in the classroom is shaping development of the brain. So by combining these interventions with brain imaging data, this is not just a picture, this is actually data that we'll get to at the end of this talk. Now, the long-term goal of this is to use neuroscience as a tool to predict learning and personalize education. So it's kind of a part of a broader idea of precision education, of using a wealth of data on individuals to make more informed predictions about where they're going to thrive in an educational setting. We're getting started on this as well, but today I'm just going to be focusing on the first part here. Okay, so the outline for this talk is the first part is going to focus on plasticity in the reading circuitry. We're going to ask the question of how much does a targeted intervention change white matter structure in children with dyslexia. Next, we're going to look at sensitive periods for white matter plasticity. We're going to ask the question, does plasticity in learning diminish with age? Finally, and if there's time, I'll probably take a poll at the end because I'm guessing I'll run out of time to do both these. We'll dive into asking the question of what's actually changing in the white matter, going beyond just looking at diffusion measures and trying to ascertain which cell types are changing and over what time course. So to start off here. So many of you might recognize this text. This is the definition of dyslexia that's used by the International Dyslexia Association. It came from a sequence of NIH panels. I'll let you all read it. And I've bolded words that I think are key here in our understanding of dyslexia, that it's a learning disability that is neurobiological in origin. Now, many people take this definition to imply that children with dyslexia have some sort of neural defect that causes persistent difficulties with reading, irrespective of intervention effort. This is an interpretation that many people take away after reading this definition of dyslexia. Now, however, there's a wealth of research on what's different in the dyslexic brain, but there's far less research on the stability or plasticity of these differences. I think this is broadly true of developmental science in general, that there are far more cross-sectional studies that look at, for example, differences between children at a certain age, many fewer longitudinal studies that look at how those differences evolve over time, and then an order of magnitude fewer intervention studies that actually intervene to ask how stable are these differences when the environment of a child has changed. So when we ask the question, what's different about the dyslexic brain? There's this is a huge literature that I'll summarize one tiny part of it in one slide. So we know that the white matter connections between the reading circuitry are crucial for skilled reading. We can look at this in one way, for example, by identifying these pathways, for example, the arcuate fasciculus in individuals, extracting some measure. I'll go into these measures a little later. This is a measure of mean diffusivity in the arcuate, and we can see correlations with reading skills. This is data from my lab as soon as, actually the first study I ran when I was at the University of Washington. And it's largely a replication of about 20 other studies that have found correlations between properties of the white matter and reading skills and group differences between dyslexic and typical readers within these same pathways. So the question that arises whenever we look at these data relating some measure of the brain to some measure of behavior is the question, are these white matter properties a static trait that constrains development? I think this is an assumption that many people make. We all know that correlation does not equal causation, of course, but if you go through the literature, you'll mostly notice that people tend to plot the measure of the brain on the x-axis, the measure of behavior on the y-axis, with some implicit assumption that the brain differences are an intrinsic difference that's potentially causing the differences in behavior. So what I think a lot of people assume is, for example, this child that's at this end of the plot might show a developmental trajectory of the white matter that looks like this. The child that's up here, the other end of the plot, will also show maybe a similar developmental trajectory, but that they'll stay different from each other over development, potentially explaining why one child is having persistent struggles with reading, whereas the other one isn't. Alternatively, we might asked the question of whether white matter properties dynamically change in response to a child's environment. So for example, in response to changes in the education, changes in the curriculum or the teacher or the way that they're being taught. So under this second model, what we might think of is that there's some typical developmental trajectory, and a child might be progressing along that developmental trajectory. And then events in their life are gonna push them one way or another and change the rate of development of specific brain circuits. So maybe the child enters preschool and development starts accelerating. Then they enter kindergarten and starts accelerating again, but maybe over the course of kindergarten, they find that they're disengaged and they're in a classroom where they don't connect with their teacher. Maybe they don't feel like they belong and development starts lagging behind. And then their parents move them to a new school where they find an engaging teacher and a curriculum that really connects with them. And we see a rapid change in development over the course of the weeks or months as they enter this more well-suited educational program for them. Under this type of model, this would mean that what we measure at a given time point might reflect a child's immediate environment as opposed to something intrinsic about their brain. So for example, we measure this kid weeks apart as they are in their old school versus a new school, we might see dramatic differences in their properties of their brain. Now, of course, this is not an either or question. It's not, is the brain, are brain properties traits or are they plastic? Of course, it's both, but this is really in a question of effect size and time scale. How much can we influence changes, for example, in the white matter and over what time scale? So that's what we did in the study I'm going to talk about right now. You can think about it as looking at this exact moment in time here in this hypothetical plot. We're going to be running a study where at the end of the school year, we enroll children in an intensive intervention program. And over the course of weeks, we're going to watch how that intervention program reshapes white matter development. So first, I want to thank the incredible research team that was running this study. This group of folks conducted 562 behavioral sessions with 250 MRI sessions, all done longitudinally in young children going through a tightly controlled education program. And none of this would be possible without all their contributions. Okay, so the study design, we had 36 children with dyslexia that were recruited for an intensive summer reading intervention program. I bolded the word intensive here because the intervention involved 4 hours a day of one-on-one training in the Lindemood Bell Seeing Stars curriculum. This was done 5 days a week for 8 weeks. So this is children working one-on-one with highly engaging, highly trained instructors, working on one of the skills that's most difficult for them and focusing on that for a good portion of their day, every day, for a good portion of their summer. For the sake of this talk, I'm not going to go into the training program in details, but you can see it's been published in a number of other papers. There's also a published manual. The training focuses on many of the building blocks of reading, such as identifying phonemes in speech, recognizing letters and visualizing letters, building up a better visual representation of letters, learning to build new words with letter combinations, and progressively decoding increasingly complex words and layering in reading fluency as well. In the study, we also had 19 control subjects going through business as usual. We scanned them over the course of the typical school year. And over the study, subjects would come into my lab every 2.5 weeks, and we would collect measures of reading accuracy, rate, fluency, and comprehension. And then we'd use diffusion MRI to measure properties of their white matter. So first, looking at the behavioral data, this is work by my former grad student, Patrick Donnelly, who's now moved on to doing some exciting stuff at Google. So this is showing data for one child. Along the x-axis is the number of hours of intervention. Along the y-axis is their Woodcock-Johnson basic reading skills. This is a age standardized measure with a population average of 100, standard deviation of 15. This is showing data for one 9-year-old child who's coming in at the floor of the test. Now, just to appreciate what that means, this is a 9-year-old. So I think this child was in 4th grade when they started this study and could only read 3- and 4-letter words. For any of you that are familiar with the typical 4th grade curriculum, you're being asked to read books, to interpret text. Much of your daily practice depends on reading. So you can imagine how difficult and even traumatizing it is for a child to be entering 4th grade, really being almost functionally illiterate. So these are data for different children. You can see this, these children start down here at the floor of the test. This child showed pretty dramatic improvement. Other children start a little higher. All these children were below average, ranging from effortful below average readers to severely dyslexic. They all showed growth over time. And this is showing the plot of the average growth for this group of children. And on average, we see children in the study going from about the 10th to the 30th percentile on measures of reading skills. This is about 1 grade level of change, or an effect size, a Cohen's d effect size of 1. We also see variability in growth, with some children increasing as much as 2 or 3 grade levels, whereas others showing more limited growth. We can look at different skills. What we see is that we see highly significant and roughly linear growth for all our various reading measures. This plot is showing data for the Woodcock-Johnson Basic Reading Skills Index. This is a measure of reading accuracy in orange, reading fluency, which includes a speeded measure of comprehension, and reading rate in green. We have a baseline period before the study began where we collected individual baseline data, and all the measures were flat over that period. And then we see roughly linear growth over the course of the study. And these are showing growth rates over here in panel C. They're showing growth rates across these different measures. One thing you can appreciate, there's significant growth on all these measures, with larger improvements for accuracy shown in orange compared to fluency or speed shown in green. We also see no improvements in math. So as a control to see whether the intervention was specific to reading versus generally improving all academic skills, we see a small decline in math. Now, no one go away saying that the intervention made math skills worse. Remember, these are standardized measures that are standardized for age. The children are getting older. This is summer vacation. They're in a reading intervention program. And I can almost guarantee that none of the children are going home and practicing math skills after spending half their day in an intensive reading intervention. This is the summer slip in math, well known. Okay, so now we can get back to our core question and ask, does the learning process change the structure of the reading circuitry? This is work done by my former postdoc, Livy Huber. We collected diffusion MRI data at 4 time points over the course of the intervention, so sampling every couple of weeks. This is I won't go a lot into the details, just give a little bit for those that are interested in the methodology. This is a multi-b-value sequence where we had 3 different b-values. We sampled 104 diffusion directions. And we used the diffusion kurtosis model to measure microstructural properties of the tracks. I'll come back to that at the end about what are the specific insights that can be gained from this type of modeling. But to give a high-level overview for those not familiar with diffusion MRI of what these measures are sensitive to. So this is showing our rendering of the archaeofasciculus, our key language and phonology pathway. If we imagine zooming in and looking at an electron micrograph of this pathway, it would look something like this. This is a reconstruction of the optic nerve from an electron micrograph. You can see all these individual axons aligned together into a bundle like this. Now, when we collect diffusion MRI data, we can think about axons as tubes that, wires that are connecting different brain regions. And we can think about water molecules shown in blue here, diffusing around and bumping into the membranes of these tubes, these axons. What I want you to appreciate is in this simulation on the left here, which is set up loosely like the child's brain, where you have axons that are relatively small, that have less insulation around them, so less myelin. There's a lot more room for the water to fuse, leading to a higher rate of diffusivity. Whereas in the simulation over here, which is more like the adult brain, where these tubes have grown larger in diameter, they're more well insulated with myelin. These are 2 major developmental changes that occur that allow these axons to conduct signals faster, to transmit more information. And we're going to see lower diffusion. So remember, lower diffusion indexes more densely packed tissue because there's more membrane, more cellular structure to block the diffusion process. So how do we make these measurements in kids over the course of the intervention? we use a software package that my lab's been developing for some years called AFQ, now called PyAFQ for those that are AFQ users. We've now released a version in Python, and all current development is happening on the Python version. Big success for the Python folks around here. This software package will take in raw diffusion MRI data. So this is showing raw data coming off the scanner. It allows you the opportunity to fit a multitude of different types of models of the diffusion process. So the tensor model that we all know well, or constrained spherical deconvolution, for example. And to use these models of the diffusion process to create, to use tractography to create a whole brain connectome of fibers, basically all the fibers that we can generate connecting different regions of the brain through the white matter. And you can select from about a dozen different tractography algorithms here. We then automatically segment these different fibers into known anatomical bundles called fascicles. So this is showing the inferior frontal occipital fasciculus identified in one individual's brain from their diffusion data. Right now, the PyAFQ package will automatically give you, I think, 28 fascicles, but that's growing by the week. It'll then take in different measures, so FA or mean diffusivity or quantitative T1 or myelin water fraction maps, and map these onto the fascicles that you identify, and then summarize data for an individual based on a vector of measurements going along the length of the fascicles. We summarize these measures of tissue properties along the length of the pathway for each individual subject. One other recent software release, I'm just going to highlight a couple software releases that people might be interested in. We developed a tool called AFQ Browser. That's a way to interactively visualize all this data, to conduct group comparisons, to share data that's completely anonymized through the web browser. You can check this out at this web link here. I know since we're on a Zoom, people might be getting distracted at this point, so now you can have some fun distractions to browse around the internet and look at some of these tools that we've made. We've made this into a whole software infrastructure, PyAFQ for processing data and identifying these pathways, AFQ Browser for visualizing, interacting, and performing exploratory data analysis, then AFQ Insight, which is a machine learning toolbox that's tailored to these data. And I've just a wonderful team of collaborators. Really want to highlight all their work that have been focusing on building this whole infrastructure. For any of you that are diffusion MRI folks, feel free to go to the GitHub site, submit issues, let us know how it's working. We're really trying to make this usable to the broad community. Okay, so now let's turn back to our main question and look at how this reading intervention program affects the structural properties of this pathway, the arcuate fasciculus. So this is showing a measure of mean diffusivity at baseline, right before these children start the intervention, the week before they start the intervention. And now this is showing another measure 2.5 weeks later after they've received on average 50 hours of intervention. What you can see is just over the course of 2 weeks, we already see a highly significant decrease in diffusion. Now remember that a decrease in diffusion generally corresponds to denser packing of tissue, basically the growth of new cells or new membranes that inhibit the diffusion process. Over the course of the rest of the intervention, we see this decrease in diffusivity persisting or growing slightly larger over time. And when we look at our control group that was not getting the intervention, these diffusion measures are stable. So we see a highly significant decrease in diffusivity in our intervention group and no change in our control group. We can look at this in a slightly more nuanced way by plotting out mean diffusivity going from the frontal lobe here, so where the arcuate fasciculus enters into Broca's area, along the length of the pathway to the temporal lobe, where it enters into posterior temporal cortex. What you can see is that this is not localized to one particular location on the pathway, but we see a change in mean diffusivity pretty much equivalent along the full trajectory of the fascicle. So what we can infer from this, since we're seeing a significant decrease in mean diffusivity, is suggesting that the intervention is actually driving the growth of new tissue, basically new cellular membranes that are inhibiting the diffusion process within the arcuate fasciculus. We can now zoom out a little bit and ask whether mechanisms of plasticity are spatially localized. So is this just about one key pathway that's critical for reading, or are they going to be distributed across a broader network of white matter connections? So we're going to look at these two pathways, the arcuate and the ILF, the ones I introduced at the beginning. These are separated by centimeters. They don't overlap at all, and they connect very different parts of the reading circuitry, though both of them are involved in reading. I'm going to show data first for one participant. This is showing one subject that was randomly selected from our sample, showing in blue the mean diffusivity values for their ILF, in green the mean diffusivity values for their arcuate across the 4 sessions. You can see this subject shows a dramatic decrease in diffusivity between session 1 and session 2 that persists over time. And the trajectory of change is almost perfectly matched for these two highly disparate pathways. We can look at 6 more subjects. You can look at these trajectories of change over time, and they're highly correlated, meaning that the changes in diffusion measures for these two pathways are tightly linked for a subject. Here's showing data for all the subjects. We can summarize this by fitting the growth rate. So looking at a linear growth rate for the ILF and the arcuate. Each point in this scatter plot shows the growth rate for an individual subject, and we're plotting them against each other. And you can see that almost down to the noise in the measurements, these two highly different pathways in the brain are showing very similar changes driven by the intervention. We can zoom out a little bit more. This was the figure I introduced at the beginning of the talk, which was actually a real data figure. Here we have shown a group of pathways in the left hemisphere. We are using a color map to represent the p-value on a linear mixed effects model, summarizing the rate of change driven by the intervention. Warm colors indicate pathways that are showing significant change when these children enter the intervention program. What you can see is that there is a large collection of pathways, the arcuate and ILF just being 2 of them. They're showing intervention-driven change by this when children enter this new education program. So to summarize, we see highly significant growth well beyond the core reading circuitry. And so this plot I showed before of the growth rates for the arcuate and the ILF is a summary of the growth rates for these two pathways here shown in this bar plot. Now what I'm going to show you is the relationship between growth across all these different pathways. So this being one cell in this matrix here showing how correlated are the growth rates for the arcuate and the ILF. And what I want you to appreciate is there's this block of pathways where this matrix is all orange and yellow, indicating that there are a network of different pathways that are showing highly correlated change, that are changing roughly at the same rate and the same time as children enter this intervention program. There's other pathways such as the cortical spinal tract. So interestingly, we do see significant changes in diffusion properties of the cortical spinal tract. However, those occur at a different rate than the arcuate fasciculus and these other pathways. There are multiple networks in the white matter that are all changing and in correlated ways. So now let's link these time courses of change in the white matter to time courses of learning in terms of looking at children's changes in reading abilities. This is showing a similar plot before where we're looking at diffusivity in the ILF, which goes down over the course of the intervention in this subject, and reading scores, which go up over the course of intervention. And you can see that for this subject, the time course of these two, a rapid increase in reading coupled to a rapid decrease in diffusivity, then flattening out over the course of the intervention, look like they're linked together for this subject. Can look at data across all these subjects and then make a scatter plot, plotting out the session-to-session change in diffusivity for this white matter pathway versus the session-to-session change in reading abilities. And what you can see is that within an individual, changes in behavior are occurring in synchrony with changes in the white matter. I put a little asterisk there to note this is a summary of an effect that is statistically significant, but you can also see that there's a lot of unexplained variance too, that some subjects show improvements in skills before they show changes in the white matter and vice versa. I think it's a really interesting point to follow up on in future work. Okay, so as an interim summary, what we see is that interventions drive large-scale changes in brain structure that occur over a rapid time scale and in synchrony with reading improvements. What we can infer from this is that white matter constitutes a highly plastic system that flexibly adapts to environmental demands. We see effect sizes ranging from cones D of 0.5 to cones D of 1.0 across different tracks. This is indexing the magnitude of change that we see over this 8-week intervention program. And let's think about those effect sizes relative to what we would see in a typical study of group differences. If you do a review of the dyslexia literature and ask, what are the usual effect sizes when a group of people with dyslexia are compared to a control group, the effect sizes usually range from about 0.2 to 0.4, maybe a little smaller. I think this is an optimistic estimate. So I want people to appreciate is that we see more change over the course of 8 weeks of an intensive intervention than we would see when we compare groups of subjects that differ dramatically in terms of their skills. I think one of the important takeaways for this is that we really should not be interpreting group differences as reflecting some static trait of the brain. That, in fact, the white matter is highly plastic and it changes rapidly as a function of a child's environment. Now, one question we might ask based on these data is whether there's a sensitive period. We might posit the hypothesis that we're seeing large effects because we're intervening during a sensitive period for white matter plasticity. So I'm going to move on now to evaluating this idea of whether the reason for the large effects here are because we're intervening during a specific period of white matter plasticity. And we're going to ask, does plasticity and learning diminish with age? So I think everyone here is probably familiar with the idea of a critical period. Here's a quote taken from a wonderful review from Worker and Hensch. They say that the classic notion of a critical period is a window, typically early in development, during which a system is open to structuring or restructuring on the basis of input from the environment. More broadly, a sensitive period is a time window during which a circuit is particularly malleable by experience. So here's a figure, a schematic figure suggesting the idea that early in development, there's some breaks that hold a system relatively static. A critical period or sensitive period will open where the circuit remains, is particularly malleable to the environment. And then as someone gets older, these biological breaks will be turned on again to fix the circuit in the way that it's already developed. We might ask, did this period correspond to early elementary school? The brain is particularly amenable to rapidly changing in response to a reading intervention. So the way we can frame this hypothesis, the way we can test it, is whether the magnitude of intervention-driven plasticity depends on the age of the subject. So the way we're going to analyze this is by looking at an age by intervention time interaction, or put in a different way, looking at the correlation between how old the subjects were when they entered this intervention and how much change we observed over the course of the intervention program. Our approach was to formalize a model. We did a power analysis based on a model of critical periods. We pre-registered this. We collected an additional cohort of subjects and the preprints online. The paper will hopefully be published soon. So let's first look at reading skill. I've divided our sample into the younger subjects shown in red, the older subjects shown in blue, the full sample with error bars is shown here in purple. You can see one thing we noticed about our sampling procedure, we just happened to sample older children who had lower reading skills. Remember, this is an age-normed measure here. But the important point when we look at intervention-driven growth, that so the main effect of intervention time, basically the p-value on our reading scores changing over time, is highly significant. Use exponential notation because there's 14 zeros before we get to the p-value. When we look at the age by time interaction, so looking at how much does the growth rate depend on the age of the subject, they are almost perfectly parallel. So in this sample, there is no relationship between how old a subject was when they entered the intervention program and how much gains they saw over the course of the intervention. In other words, the red line and the blue line are almost perfectly parallel. We can now look at the same plot for the white matter. Now, to reduce the high dimensional space of all these many white matter tracks to a single measure, we used PCA for dimensionality reduction. This is going to capture the correlated change that we see across this spatially distributed network. And what I'm plotting here is the session to session change for all the subjects in the first principal component of the white matter. And we see a main effect of intervention time, P of 0.002. show highly significant change in mean diffusivity, as we already saw before, over the course of this intervention. When we compare the younger subjects with the older subjects, and the arrows on the right side here are showing how much the older in blue versus younger in red, how much they change over the course of the intervention program. We can compare those, and you can see that there's no support for diminished plasticity with age. We see almost the same amount of change on average in the older versus younger subjects here. Younger subjects do start with higher mean diffusivity, as would be expected. So the age by time interaction was not significant, and the correlation between each individual's age and their mean diffusivity change was R equals 0.03. So essentially no relationship between age and the amount of plasticity we observed. So we can look at this in one more way. What I'm going to show here are the large scale changes in diffusivity that we see. So this color map shown going from black to yellow is the p-value from a linear mixed effects model, looking at each point in the white matter and how much it changes over the course of the intervention program on average. So you can see that there's as we showed in that other slide, that was average for different white matter pathways. Throughout the white matter, we see highly distributed effects where there's a lot of different areas of the white matter that are showing highly significant changes over the intervention. We can now look at, within the same model, the age by time interaction that's shown down here. And what you can appreciate is that there's really nowhere in the white matter, even if we don't correct for multiple comparisons, we don't see anywhere in the white matter where there's a dependency on age for the amount of change we see. Maybe there's a couple isolated regions, but not broadly across the white matter. So the question of, is there a sensitive period? I want to emphasize that this is inferences should not be taken as a general one, not yet, but for the intensive one-on-one intervention program that we used here, we saw neither white matter plasticity nor reading growth rates diminish between first grade and middle school. In other words, the older children were showing the same amount of change in reading skills and the same amount of change in white matter as the younger children. I don't want to say this is a general phenomenon, but we think it's true, at least for this one-on-one intervention program, where the intervention, because it was one-on-one, because it was highly intensive, could adapt to the needs of each individual participant. Okay, so I think I got a couple more minutes. May I'll pause here if there's any questions. And if there aren't, then I'll go to the last section, looking at the neurobiology of white matter plasticity and trying to ask what's actually changing.
Excuse me, may I ask a question, or am I sort of interrupting?
No, great. Thank you, Fernando.
Hey, Jason. So I was wondering, I mean, you were very cautious on stating it, the fact that, you know, it's between kind of middle school and the younger children there was no change. So the question then addressed is more than if there is a sensitive period, is if it tells more if within the scale you see differences.
Mm-hmm.
So do you see what if it goes? It's more like if there are differences within this specific scale, but you know there's a very long time point.
Very good point. Yeah, let me see if I have a hidden slide here that. Let me go to this. Okay, so before doing this, as I said, we did some modeling based on our previous paper and pre-registered this. And what we did is we posited a model of what a sensitive period would look like. Simple model is a Gaussian where let's just say the peak is centered when children start elementary school. It's a fine assumption, I think. And the sigma, so like the width of the Gaussian, is the kind of width of the sensitive period. And Fernando, I think what you're saying is that we shouldn't conclude there isn't a sensitive period. Maybe there is one that doesn't close until later. So like looking at this sensitive period model shown in bright red here, this is with a sigma of 6. This would be saying that the sensitive period doesn't close until age 19. Maybe it doesn't close until even later.
Yeah.
And then what we simulated here, so the age range of our data is shown in this gray box. So we had data up until age 13. And what I'm showing over here is a power analysis, which is showing what would be our likelihood of detecting an effect if the true sensitive period closed by this age. So this is our age range here. And so if we have a sensitive period that closed by age 14, we'd have an 85% probability of being able to detect that. We had a sensitive period that closed by age 13, we'd have about a 90% probability of being able to detect that. So I think one of the important points is, like, we're not saying that there isn't a sensitive period, just that there isn't one that closes during this elementary school period. Beyond that, we wouldn't have the power to actually be able to detect that. So maybe I'll take one more question if there's another question. Otherwise, I'll dive into the last part of the talk. Okay. So what's the underlying biology of rapid white matter plasticities? And I emphasize in human because I think as a field, we know a lot about the animal brain. We know a lot less about the human brain because we can only observe the human brain changing over time, non-invasively, of course. And when we think about what's in our imaging voxel, here's a schematic of a voxel. We know that actually 50% of the voxel is filled with glial cells. So astrocytes and oligodendrocytes, microglia. What we usually think of in the white matter are axons, of course. But even axons themselves, you have the axon, you have the myelin, there's a lot of different tissue here. All these types of tissue impact Gaussian diffusion. So meaning when we use a tensor model, DTI, the diffusion tensor model, that that model will be affected similarly by all these different types of tissue that are mixed within our imaging voxels. However, there's been a lot of beautiful work. We've been using some by Els Veermans, who's at NYU, of positing biophysical models that capitalize on non-Gaussian diffusion to isolate properties such as axon water fraction. What do I mean by axon water fraction? I mean the fraction of water that's trapped within axons versus the total amount of water within the voxel. So you can think of this as a measure basically like how much of this voxel is filled with axons versus just glial cells and water. Before going into this, I want to emphasize, all these models depend on assumptions. It's an active area of research of testing and validating different model assumptions, the extent to which they're true in terms of the underlying biology, the extent to which we can recover them based on models of the diffusion process. And it's a rapidly changing field. I think that these models should be interpreted cautiously, but we can gain additional information about tissue biology. So this is showing a axon water fraction map derived from our data. For those collecting diffusion data, these are measures you can derive once you collect multiple B values, basically specific acquisitions that have multiple B values in a lot of diffusion directions. Now, interestingly, or importantly, sorry, is these measures, once they're processed in the correct way, for example, using the PyAFQ software package, they can be highly reliable. So this is showing the scan rescan reliability of axon water fraction for the arcuate fasciculus. We have a reliability of 0.9. So this is a highly reliable measure, actually more reliable than most of our clinical reading assessments. I'm going to skip over this. This is showing the reliability of various measures across different tracks. This is work from Elizabeth Huber, examining what you need to do to actually make these more complicated models reliable. Big takeaway, though, is that on average, these parameters such as axon water fraction or orientation dispersion index has similar reliability to FA, a classic diffusion property, and hover around 0.8 depending on the track and the acquisition protocol. So what hypotheses are consistent based on mouse models? What do we know about white matter plasticity coming from model organisms? So wonderful work by Aaron Gibson in 2014, one of my favorite papers of the decade, where they performed optogenetic stimulation of mouse motor cortex. This allowed them to then systematically chart the cell types that were changing in the white matter as a function of time after basically injecting stimulation, so injecting kind of fake neural activity or real neural activity, but manipulated by the researchers through motor cortex. And one of the first things they see that within both cortex and white matter, within 3 hours of this stimulated motor activity, they see proliferation of oligodendrocyte precursor cells. And over the course of 4 weeks, they see a large growth of new oligodendrocyte precursor cells. So rapidly, the takeaway for those that don't think a lot about neurobiology is that as circuits are being active in a new way, you see actually the growth, the proliferation of new oligodendrocyte cells. These are the cells that will eventually go on to produce myelin, but these cells aren't necessarily producing myelin at this time point, and are rapidly growing as a result of changes in electrical activity within a brain circuit. So when we think about our model of a white matter voxel, we can think about a couple things that might be changing. One might be an increase in axon caliber and myelination. So you imagine these axons growing additional wraps of myelin over this 8-week intervention. I think this is what most people assume is occurring when we see changes in white matter diffusion properties. We could pick up on this in theory with this axon water fraction measure. This is showing the average amount of change for all these different pathways. The average amount of change in axon water fraction for 20 different white matter pathways in the brain. And we don't see any significant changes in axon water fraction over the course of this intervention. Another biological mechanism that might be occurring is glial cell proliferation. So similar to the work in mice done by Erin Gibson here at Stanford, where she observed the proliferation of oligodendrocytes in response to new activity within motor cortex. In this schematic here, I've drawn new oligodendrocytes within this imaging voxel. And we can theoretically pick up on this type of change based on another parameter called hindered mean diffusivity. This is the rate of water diffusion in the extra-axonal space. So a model of how rapidly water is diffusing outside of the axons. And here we do see a highly significant change in extracellular water diffusion, which is consistent with the idea that what we're actually observing over this rapid timescale isn't the growth of new myelin, but is the proliferation of glial cells that may eventually go on to create myelin. Now, I want to say this is an interpretation of some complex modeling, but I think that these models really do have the power to inform new hypotheses about the sequence of changes that are going on in the brain. And I want to end by highlighting a quote from the late Ben Barisa, one of my scientific heroes. Ben wrote that glial cells are critical participants in every major aspect of brain development, function, and disease. I think are a critical understudied cell type, particularly in human neuroscience, where a lot of times we, because we can't directly measure them, we forget about the complexity of the cellular organization of the human brain. So in summary of this last section, the link between white matter plasticity and learning involves a sequence of changes in multiple cell types and over multiple timescales. We should think beyond just myelin, and hopefully with improvements in MRI acquisition and improvements in biophysical modeling, we can keep pushing forward in understanding the link between changes in children's learning abilities and the neurobiology of their brains. So I want to end by thanking all of the incredible people that have been working in my lab on these various projects, and of course, our funding sources and the institutions that support us. So thank you all so much, and I'm happy to take questions.
Thank you, Jason. That was a fantastic tour de force. Does anyone have questions? Do you want to unmute yourself since we can't see everyone on the screen? Please feel free to unmute yourself.
So I see a question in the chat from Ted Tereski, who's asking.
Do you want me to read it, or you want to read it?
Oh yeah,
some
you
people
can read
might
it.
be
That'd
just on the phone. Some people are on the phone, so I will read it, or driving. So Ted Tereski asks, thank you so much for all the exceptional tools and for the great talk, Jason. I thought I heard you mention today that future developments to AFQ will be for Pi AFQ. Does this mean the developments to the MATLAB implementation for AFQ will be discontinued? Totally fine, just curious.
Yeah, sorry to all the MATLAB users. Yeah, most of our MATLAB tools were slowly deprecating. They still work. I still fix things that are simple to fix, but as many of the young people here probably feel, the field is really moving towards Python. The field of data science, computer science have largely embraced Python. So we're trying to embrace that too. So all the software developers that are working on these open source tools, we've made a switch to doing them all in Python. One of the big benefits from that is that we get to interface with some other just wonderful community software projects. So the diffusion imaging in Python, or DIPy package from Eleftherios Galafalatis at IU, Indiana University. Great package for processing diffusion data. And now we're able to interoperate with that, which allows us to, within PyFQ, use any of the 20 or so different modeling approaches or tractography algorithms that that are available there. So we are switching to Python for all these, also to operate with machine learning toolboxes like scikit-learn. We'll continue supporting the MATLAB version as long as, well, I'm the only one that supports it. So it's whenever I have time, usually at night after kids have gone to bed. So.
Remember those days. All right, so Lori Cutting says, thank you. It's a great, wonderful talk. Andrea Burgess says, hi, Jason, really great talk. In the second part of your talk, could you clarify if the older or younger children show differences in responsiveness to reading intervention, or were they similar?
Yep, there was no relationship between age and response to intervention in this study. Now that contradicts some just wonderful, wonderful other work from Marianne Wolf and Maureen Lovett, much larger scale study showing that interventions are particularly effective in first and second grade students with diminished efficacy. There's still large effect sizes in their intervention, a wonderful intervention approach. As children get older, highlighting the idea that we really do want to intervene early. In this case, one thing though different about this work, this was a highly intensive one-on-one intervention, really allows the intervener to adapt very specifically to the needs of the specific child they're working with. The upside of this intervention approach is it's great for doing a scientific study like this. We have large effect sizes to work with. So we have behavioral effect sizes of about Cohen's d of 1, which really makes it feasible to link change in behavior to change in the brain. The downside is obviously one-on-one intensive interventions aren't scalable given our country's current economic model of education to public school districts. So I don't want anyone to take away that, well, because of these data, we don't need to intervene early. And the other way you should look at this is, given that even if an intervention is equally effective at different ages, why should a child have to suffer through school and find themselves so far behind over years because they're being denied the opportunity to an intervention? For other intervention programs, there have been relationships between the age at which they're delivered and the growth that's seen.
Great, thank you. Do you want to, until someone has okay, you said that it was going to be a pain to pull it back up. So in case someone has questions related to the slides, let's keep it up. I was going to ask you to take it down, but given how hard it was to put it up,
so I'll take down the slides, but I will go back if anyone wants them.
All right, great. So Tracy Santani says, I'm going to skip the compliments here. I was wondering if you could speculate a bit about whether you think this potential proliferation of glial cells might occur differently in those with dyslexia compared to their typically developing peers. Is this a potential neurobiological differences we should be considering?
Yeah, totally. Great question. I'm very interested in it. I have zero data beyond pure speculation. So rather than just purely speculating, I'll say, for myself, that's one of the directions I'm deeply interested in, is trying to leverage new data acquisitions and new modeling approaches to try to understand mechanisms of learning with a lot more depth beyond just saying, well, there's a change in reading skills, there's a change in diffusion. I want to be able to say, well, what were the sequence of events that led to the change in diffusion? And Tracy, to your point, I think there's a basic science hope. Doing good quality science is just important, and understanding these things, I think we all think is important and useful for the field. And then hopefully there might be practical implications. Maybe as we dive deeper into the modeling, we'll be able to better understand why some children learn so much easier in one program than another, for example. So I have no data to answer that, but I think it's a really great direction to go. I'm going to call on Tamar Green because I see a raised hand.
Hi, thank you, Jason. It's the first time I hear you talk, although we are in the same institute, but a wonderful talk. I have a few questions. One is, you mentioned how improve the kids in the one-on-one intervention, but is there like a ceiling to that? Like how much they can improve and how, like, who are the kids who are going to meet the, and
yeah, I mean, another way you could say that, so over 8 weeks, we saw linear growth. Like the nice thing, we sampled very regularly, so we could see that growth and all the reading abilities we measured, roughly linear, meaning you get the same improvement for each hour of intervention. At some point that has, you can't get to infinite reading skills. So at some point that's going to flatten off. And
no, but flatten on the on like average level of reading for age, or flatten below?
No, it definitely flattens above that. I mean, if you as a parent want to keep paying for a intensive intervention program to get your kid way above grade level, like you certainly could. I think there's probably a diminished return in terms of the effort and cost to benefit. At some point, like you probably want to work on other skills. And I think it's also, another way to think about this is dose response curves. And in the Donnelly, I think it was 2019 paper, it's published in Frontiers in Psychology. Patrick Donnelly did some work of like modeling dose response functions relating, hours of intervention to growth rates. And there's a lot more detail in there.
And my just quick follow-up question in terms of brain development, do you expect changes, or is there any data? Are the changes on these identified tracts that are, identified in dyslexia, or would you expect like compensating tracts to develop?
Yeah, I think our field has kind of misguided itself in some ways, and that we, as psychologists, we like put these labels on different parts of the brain as like, core or compensatory or unrelated. And sometimes that in some ways that's really useful organizing structure for thinking about things. And though sometimes when we look at the data, it doesn't carve as neatly along these like labeled, the joints that we've labeled in the brain. I'll pull back up the image that will clarify what I'm talking about. Well, okay, this is a good one. So so this slide here is showing the amount of plasticity, the growth rate as a function of the intervention across all of the white matter. And it's really distributed over a lot of the brain. I think it's hard for me to find an organizing principle of saying like it's the core reading circuitry or it's the compensatory tracts. Like it, I think in this case, maybe a more useful way to think about it is like thinking about what it's like for a child to enter one of these intervention programs. If you imagine a 9 or 10-year-old kid who's been struggling reading for years, many of whom school has become a stressful, anxiety-producing environment. Many of them, struggle to connect with their teachers because they can feel, you can feel like an outsider in school if you're really struggling to do these things other kids are doing. Then suddenly you enter this intervention where you're being validated, you're working with someone who's highly engaged and meeting you at your needs, that's validating the experience you've had and that you feel that you're learning and making progress, maybe for the first time. There's so much more going on than just learning to read in terms of like how entering an intervention program like this changes a child's day-to-day experience. And I think that we're seeing, this is purely speculation, obviously, because I haven't run RCTs where we manipulate like how much the intervention person or the intervener engages with the child or makes them feel validated. I don't know if those would even be ethical studies to do. I wouldn't want to assign kids to the, the unvalidated condition there. So purely speculatively, we're measuring a lot of those other impacts that are related to reading development. And those are really important aspects of an intervention for accelerating reading growth. but they're not what we think of as like, you know, the core curriculum, like the skill that's being drilled on. So I think when we look at brain plasticity, particularly when it's brain structure, right, we're going to see the changes related to this dramatic change in the daily environment, not just the changes that are linked to improvements in reading. Yeah, thanks for that question.
All right, there's a couple more questions, but I'm going to selfishly ask one. I think it's related to this one when you were talking about large, and I think there's a couple others that are related, but so I'm trying to kind of reconcile or sort of integrate your kind of comments between, I think for reading, you saw some specificity and improvement for this reading intervention compared to math. And then on the other hand, also you're talking about brain and different fibers being very coupled in terms of the directionality, which all sort of makes sense. But then you see these large scale changes that seems to be sort of almost everywhere in the brain. But then, so then of course, these different white matter tracts that shows similar kind of trends should have some kind of behavioral implications as well. So I'm trying to think about chicken or egg story, and then also like how you're kind of connecting between specificity and behavior, and then kind of these diffusivity, global diffusivity changes that seems to have kind of other cognitively
really
associated
great question. Really great question. That's something that I've been grappling with a lot myself over these years since we've switched to doing kind of like interventions being the lab methodology. So in terms of behavior we measured, yes, like it was specific to reading, but there's also a lot of many other unmeasured aspects of behavior. So in this first study, we didn't, you know, ask children about their sense of belonging and their sense of self-efficacy and their sense, you know, their mindset relative to reading. You know, did children come in with a fixed mindset where they thought that, you know, they were bad at reading, they'd never be good, and then did they, over the course of the intervention, change their mindset to a growth mindset where they thought if they worked hard, they could get better at reading? You know, there's all these, you know, nuances of behavior and cognition that, you know, because we can't measure infinite things, we focus on like the things that are hypothesized in, or, you know, the focus for interventions. There's most certainly a lot of other, particularly like the children's self-concept and how they think about themselves, that are their emotions that are changing over these intervention programs. Now you layer on a curriculum, and the skill that they were being taught within this like broader context of like a highly engaging academic environment was reading. So they didn't, you know, just feeling engaged and feeling affinity towards a teacher, you're not going to learn algebra if you're not being taught algebra, right? You're not. So, you know, I think when I think about like the future of this field and where we should be going, you know, I think, you know, thinking about a lot of the mediators to learning is really important. Like how much are these kind of I don't know what to call them, like, you know, children's self-concepts, are they, you know, their mindsets, to what extent are they key mediators for learning? And maybe a lot of the plasticity we're measuring are related to those changes in like mindset, for example, as opposed to the exact skill that's being taught. And you know, I don't have an answer, but that's one thing that we're working on in current studies. We have an intervention going on right now where we're measuring a lot of these other factors. We're also doing longer term follow-up, so at 6 months and 12 months. The other thing you could have asked is like, well, what happens a month after they exit the intervention program? Do their reading skills keep growing? Does it go back to baseline? Does it stay where they are and they keep needing, I don't know, to take an analogy from our current pandemic, we keep needing another booster and another booster of intervention to keep, you know, getting back to where we need to be? Those are other directions we're trying to figure out now.
Great, thank you. I think just to follow up on it, it's interesting because if you see behavioral compensation studies in behavior, then they all point to language because that's what they target. Right, so you only see what you measure and where you're shining the light, and that's what you see. And then when you do imaging and whole brain approaches, and you kind of do the reverse kind of functionality and trying to locate the functions based on the tracts or brain regions, then you see a lot more in terms of cognitive and other things that people have not usually measured before.
Totally, so that's a great point. You know, and you think about differences between anatomical imaging and functional imaging, and like, you know, over a reading intervention, if you're doing task-based fMRI, you're most certainly going to use some kind of reading task that activates specific regions that are involved in reading. And so the only changes you're going to see are in those regions that are activated by the task. But that doesn't mean that's the only thing in the brain changing. If you decided to do a reading intervention and you were a researcher studying emotion, and you just said, I don't really care about the reading score, but I'm going to look at, like, you know, emotion changes and the brain's emotion circuitry. You know, maybe you, Fumiko, as you were saying, maybe you discover really interesting changes in, you know, these circuits that we don't think of as being core, but you know, maybe they are. And we need a more integrated approach and how to get there. I don't know, it's like more data, but more data takes more time. I don't know.
Great, thank you. I know it's kind of pushing time, but I just wanted to, if people have to go, I know some people have left already, but please feel free to go and don't feel like you're being rude if you left before Jason. But I just want to make sure, do you have 5 more minutes? There's 3 more questions.
I do, yeah, I can
here.
stick around for 5 or 10 more minutes.
Okay, great, thank you. So one question is from Colleen Mercado, and she says, or I'm sorry, well, I don't know the gender, I apologize. So Colleen says, in a practical sense, are all reading programs that address dyslexia going to see similar outcomes in brain plasticity, or are there some methodologies that were used in your study? I think this is, I feel like I know what you're going to say, but is there aspects in your study that you know you want to be replicated and that used in practice?
Yeah, no, that's a great question. So as you might guess, one of the things I'm really interested in is like what are the aspects? You know, I don't know. I kind of wear 2 hats. As a neuroscientist and as a education scholar, and you know, I'm really interested in thinking of doing things that have impact on learning and impact on the classroom. But I'm also deeply interested in the basic science of brain plasticity, and you know, understanding what are the different aspects of the intervention that are critical for driving plasticity is a key scientific interest of mine. Now, it turns out, as I look out at a bunch of text representing different people who are on this Zoom call, I think we can all appreciate how much the world has changed since when we did this study. And it's really hard to do a lot of these things now that we're switching from in-person to Zoom and masks to not masks, back to masks. You know, I worry it's just gonna be harder to figure out kind of a lot of these aspects of the learning environment, how they impact brain plasticity, because our learning environments right now are so unstable. We're running this study right now, the follow-up, and we're looking at a lot of these factors that are brought up, but we're also having to change a lot of things month to month or year to year based on, you know, caseloads, based on what's going on with this pandemic. So
Great.
An unsatisfying answer, but that's where things stand.
All right. So I think Kulpreet, Chima, if you're still on the call, it seems like your 2 questions were answered. If you feel like they were not answered, please just type in again, you missed it, or they were not answered. But I think it's about self-efficacy and these things, if you're measuring those. And Jason said that he is measuring those. And then if you're doing follow-up, and you are doing follow-up in these children, and I think you're seeing some stability. Is that what you said, Jason?
Mm-hmm. Yeah.
Okay, so then Su Yu Cheng's question is, how are you measuring growth rate? Is this diffusivity? And then also, she said she may have missed this. And then also, did you find anything with FA? So fractional.
Yeah, FA and mean diffusivity looked pretty similar. They're the reciprocal of each other. Increase in FA, decrease in MD. For measuring growth rates, we're just using regression to fit linear growth rates to the individual's data. We've also looked at, you know, more complex nonlinearities, but there really wasn't, and did model comparisons, there really wasn't any support for nonlinearities, probably a sample size issue. I think it'd be really interesting, by the way, to think about, well, where we started with this is wanting to look at which is leading and which is lagging changes in behavior versus changes in white matter. And that's something that we're still thinking about modeling, but we need to get a larger sample to tackle that.
Great, thank you. I think just one final question here is just out of curiosity. This is from Shannon Kushchel. She says, or Shannon says, what encompassed your business as usual activities?
Yeah, they are very variable. Business as usual was the typical school year. So we made the decision to scan the intervention group over the summer and then compare that to 8 weeks of the school year. There's like strengths and weaknesses to that. Obviously, the ideal would have been random assignment, but it's pretty well, impossible really to do random assignment with the intensity of this type of intervention program, because we couldn't like find a lot more like details there. So yeah.
Great, thank you. I think you got to cover all the questions, and